Good morning. This is the second video of our histology series, compound epithelium. Already we have discussed the simple epithelium. Those who have not seen, please see the video, then continue with this video. Next, compound epithelium. Compound epithelium is otherwise called as stratified epithelium. Stratification means multiple layers. Stratification means multiple layers. So the cells are arranged in multiple layers. Cells are arranged in multiple layers. Depending on the shape of the outermost layer. Depending on the shape of the outermost layer of the cells, the compound epithelium is classified under the following types. That is stratified, squamous, stratified, cuboid, stratified, columnar, and finally there is a special type of stratified epithelium called translational epithelium. Translational epithelium, this is otherwise called as urothelium. Okay. Now the stratified squamous epithelium is again classified into two variants, depending on presence of keratin layer and absence of keratin layer. When the stratified squamous epithelium contains keratin layer, it is called stratified squamous keratinized epithelium. It is called stratified squamous keratinized epithelium. When the stratified squamous layer or when the stratified squamous epithelium doesn't contain keratin layer on top, it is called stratified squamous non-keratinized epithelium. So this keratin layer is a protective layer. This is mainly present in skin. So stratified squamous keratinized epithelium is present in skin, and stratified squamous non-keratinized layer is epithelium is present in parts of esophagus. Now let's learn how to draw stratified squamous keratinized epithelium. As the name implies, squamous, that means the topmost layer of the cell should be squamous, stratified, so there should be multiple layers. So the final, above the squamous layer, that is the topmost squamous layer, we have the keratin, which will be lining the top of the final layer. So first, let us see how to draw stratified squamous epithelium. Draw the basement membrane first. Then on top of the basement membrane, draw the first layer of cells, that will be usually columnar or sometimes it may be cuboidal. So this is the first layer of cells. Like already we have drawn the simple epithelium. If you have not watched the video of how to draw the simple epithelium, just watch the previous video. So this is how we draw the columnar epithelium. Draw the cytoplasm for it. Then draw the nucleus, elongated nucleus towards the base. So each and every feature of cells are very important. So most of the students go wrong while drawing the nucleus. Next, we should draw multiple layers of polygonal cells. So it will have, polygonal means it will have multiple facets. The cells should have multiple facets and they should not have much gaps. So draw few layers of these polyhedral or polygonal cells, polyhedral cells. Okay, multiple layers of polyhedral cells. You can draw up to 3 or 4 layers of, minimum 3 or 4 layers of polyhedral cells. After that, the final layer should have the squamous epithelium. Now to complete the final layer with squamous epithelium, which is just flat cells. Just draw the flat cells on top of the polyhedral cells. So these layer also should be multiple. There should be multiple layers of squamous cells. At least 3 or 4 layers of squamous cells we can draw here. Now what we can do is we can just shade the cytoplasm for all the cells just like this. Next we can mark the nucleus. For the polygonal cells, we have to draw the rounded nucleus at the center. So this is how we draw the nucleus of polygonal cell. Next, draw the nucleus of the squamous cells. This is also rounded nucleus. The nucleus should be at the bulged portion of the cell. So our stratified squamous epithelium is completed. So if you draw only this much, it is called stratified squamous epithelium. Next, how to draw stratified squamous keratinized epithelium. On top of this last layer of squamous epithelium, just shade without leaving gap. So this shade represents the keratin layer. So this shade is nothing but the keratin layer. So now the epithelium becomes stratified squamous keratinized epithelium. So this is how we draw stratified squamous keratinized and non-keratinized epithelium. Stratified squamous epithelium is present in esophagus. Stratified squamous keratinized epithelium is found in skin. Next, how to draw stratified cuboidal epithelium. For stratified cuboidal epithelium, the process is same. Draw the basement membrane. Then on top of the basement membrane, draw the cells. Here we can draw multiple layers of cuboidal cells, one after other. Usually 2 or more layers of cuboidal cells can be drawn. So you know how to draw the simple cuboidal epithelium. So here what we have to do is we have to draw multiple layers of cuboidal epithelium. Usually 2 or more layers of cuboidal epithelium in stratified cuboidal epithelium. So the process is the same. Just draw the cuboidal epithelium, then shade the cytoplasm. Then you can note down the nucleus. Can place the nucleus exactly at the center of each cells. So this is how you complete stratified cuboidal epithelium. Where do we find stratified cuboidal epithelium? Usually the stratified cuboidal epithelium is found in the ducts of glands. So ducts of some of the glands, we can see the cuboidal, stratified cuboidal epithelium. Next, how to draw stratified columnar epithelium. Here you can draw multiple layers of columnar cells. Draw the basement membrane. Draw the first layer of columnar cells on top of the basement membrane. Then following which you can draw the next layer of columnar cells. So this type of epithelium is very rare in humans. Usually it is found only in places of some large excretory ducts and in places where places such as conjunctiva and in some parts of anal canal. So only these places you can see this type of epithelium, the stratified columnar epithelium. This is the same way. First draw the cells, then shade the cytoplasm, then you can place the nucleus. You know where the nucleus of the columnar cells will be. It will be basally placed elongated nucleus. So again, don't make mistake in drawing the nucleus. So this is how we draw the stratified columnar epithelium. The next type of epithelium is translational epithelium. What is this translational epithelium? This is otherwise called urothelium. So this is a special type of epithelium. Its main purpose is protection in the urinary tracts, especially in what? The ureter and also in the urinary bladder. So what is the arrangement of this epithelium? So just like how we drew the stratified squamous epithelium, here also there will be a basement membrane followed by tall columnar cells. So the basal layer will be columnar cells. So here also we are drawing the basal layer as columnar cells. Followed by columnar cells, the next layer will be globular cells or pear-shaped cells. The cells will be pear-shaped, something like this. Cells are pear-shaped. So you should take care that the cells will be almost the same size like that of the columnar cells, not more than that. Just draw the pear-shaped cells. Don't make sure that there won't be much gap between the cells. So we can draw few layers of this pear-shaped cells. These cells will help in stretching of the cavities, like the ureter and urinary bladder has the capacity of storing more urine and for the transport of urine. So it has to stretch constantly. So these cells will help in stretching of this epithelium. So when it stretches, the epithelium becomes very much flatter. Okay, then after making a few layers of these type of cells, that is pear-shaped cells, we have to draw a large umbrella-shaped cells. So the size of the cell is very large and it will be covering at least 3 or 4 cells. It will be covering at least, see, it will be overlapping at least 2 to 3 cells at least. Okay, so these are umbrella-shaped cells. Now, how to draw nucleus? So already you know how to draw nucleus for the columnar cells. So it will be elongated basally placed nucleus. So first let us shade the cytoplasm. Now we have shaded the cytoplasm. After shading the cytoplasm, let's draw the nucleus. So elongated basally placed nucleus for the columnar cells. The next, the pear-shaped cells will have rounded nucleus. So rounded nucleus at the widest portion. So it will be mostly towards the center only. So you don't need not worry. So just you can draw the rounded nucleus towards the widest portion of the widest part of the cell for the pear-shaped cells. Then for the umbrella-shaped cells, the size of the nucleus will be larger and it will be towards the center. And you can use 2 nucleus, or you can place 2 nucleus for some of the cells, because some of the cells will be multinucleated. So some of the cells will be multinucleated, where you can place more than 1 nucleus. So this is how we draw translational epithelium. So with this we have completed the epithelium. Hope this video is useful for you. Share with your friends so that it will be helpful for them too. Thank you.